Hello and welcome to this A-Level Chemistry explanation video about amino acids, proteins and DNA. In this video we'll explore how these small but essential molecules link together to form complex structures that carry out the vital processes of life. Amino acids are the building blocks of life. And although there are only around 20 of them, they combine in countless ways to create the huge variety of proteins found in living things. Each amino acid has the same basic structure, a central carbon atom with 4 covalent bonds. The first bond is to a hydrogen atom, and the second is to an amino group, or more formally, the amine functional group NH2. The third covalent bond goes to a carboxylic acid functional group, sometimes called a carboxyl group, COOH. What makes each amino acid unique is the R group, or sometimes it's referred to as the side chain. And these are different for every single amino acid, and they can vary greatly. Since amino acids always contain the amino group and the carboxyl group, and sometimes additional features in the R group, they can show both acidic and basic properties, as we'll see later in this video. You need to know the general structure and bonding in amino acids, but you don't need to memorize any examples. Many exam boards, for instance AQA, include examples of amino acids on a datasheet inserted into the paper. Sometimes examples can be given to you as part of the question itself. The AQA datasheet, for example, includes 6 amino acids: alanine, where the R group is a methyl group; aspartic acid, where the side chain is a CH2 followed by a second carboxylic acid group; and cysteine, where we have a CH2 and then sulfur bonded to an atom of hydrogen. In lysine, we have a long carbon chain and then a second amino group to go with the first in the general structure. Phenylalanine has a benzene ring connected as part of its side chain, and serine has a CH2 and then an alcohol functional group. So as you can see from these examples, the R group or side chain is what makes each amino acid unique. These side chains determine how the amino acid behaves, whether it's polar or non-polar, acidic or basic, and whether it's hydrophilic or hydrophobic. And all of that plays a huge role in how proteins fold and function. All amino acids found in proteins are 2-amino carboxylic acids. And this is because the carboxylic acid group has the highest priority, and so the carbon atom it contains is given the number 1. And the amino group is therefore always attached to the second carbon in the chain, carbon number 2. A common question for 1 mark in an exam is to give the IUPAC name for a particular amino acid. And so, for instance, with alanine, we identify the carboxylic acid and know that this is carbon number 1. Because it's a carboxylic acid, its name will end in anoic acid. We need to find out how long the carbon chain is, and you can see that this chain contains 3 carbon atoms, and so this is propanoic acid, but we have the amino group attached to carbon number 2, and so this is 2-amino propanoic acid. For serine, which I'm showing here, we can again see the carboxylic acid group, and in fact, again, we have 3 carbon atoms in the chain, so this is propanoic acid, but we have an amino group and a hydroxy group this time. And so first of all, we need to assign the alphabetical order. Amino will come before hydroxy. And so this amino group is attached to the second carbon atom, and the hydroxy group is attached to the third carbon atom. And so this will be 2-amino, 3-hydroxy propanoic acid. As one last example, if we take a look at lysine, the carboxylic acid has a carbon chain length of 6, and so this will be hexanoic acid. This time we have two amino groups attached to this chain. They are in position number 2 and position number 6. And so this would be called 2,6-diamino hexanoic acid. Amino acids exist as Zwitterions at room temperature, and this term comes from the German word for two, which is zwei. And so we're saying that the amino acid is literally a double ion. But it isn't just any two charged atoms within the molecule, as you'll see when we look at some examples. For instance, with alanine, which in full looks like this, but actually at room temperature it exists as a Zwitterion, where the carboxylic acid group is deprotonated, meaning that it has lost an H+ ion, and so is negatively charged. And the amine group has been protonated, so it has gained an H+ ion, as you can see from this diagram here. And it's specifically these two regions which makes the amino acid a Zwitterion. Any other two places instead, and it's not a Zwitterion. You need to be able to draw the structure of a Zwitterion from a given amino acid. And so if we look at a second example of serine, the carboxylic acid group is deprotonated and becomes COO-, and the amine group gains a hydrogen ion, and so it becomes NH3+. Amino acids are solids at room temperature because even though they have an overall charge of 0, in other words, they are neutral, their structure is an ionic lattice, and so they are crystalline solids. And this is due to the ionic bonding, with electrostatic attractions between the positive part of one Zwitterion and the negative part of another. And so this means that they have a melting point above room temperature. Most amino acids contain an asymmetric carbon atom, sometimes called a chiral carbon atom, and this means that they will be optically active. Remember, an asymmetric carbon atom has 4 different groups attached to it, which applies to all 2 amino acids, except for glycine, where the R group is a hydrogen atom. Exam questions sometimes show a molecule, for instance serine, and command you to circle the asymmetric carbon atom. And so that would be this second carbon atom that I'm showing circled here. Amino acids then exist as pairs of stereoisomers, which are defined as molecules with the same structural formula but a different spatial arrangement. We can show that by drawing one molecule of serine, showing the tetrahedral arrangement around this second carbon atom here, and then drawing the second enantiomer as the non-superimposable mirror image of the first. These 2 enantiomers can be distinguished using plane polarized light, which works because the 2 enantiomers would rotate light equally, but in opposite directions. Amino acids are amphoteric, and this means that they're able to react as a base or an acid. In an exam, a common question is to ask what structure is formed from a particular amino acid, for example serine, at pH 3, or they might instead say in acidic solution. Well, in both of these cases, the hydrogen ion concentration is high, and this means that the excess hydrogen ions in the solution will react with the functional groups that can act as bases, which are, of course, hydrogen ion acceptors. And so for serine, in acidic solution with an excess of hydrogen ions, the amino group uses its lone pair to accept a proton, and we make this structure here, where the amino group has acted as a base and become protonated, and we're showing this as an NH3 group that is now positively charged. It's worth noting that amino acids can have acidic or basic R groups. And so for a question like this, it's important to check for these side chains first. And if a question had asked about lysine, which has an amine group as part of its side chain, the excess hydrogen ions would protonate any groups that can accept a proton. And this means that both of the amino groups would be protonated in acidic solution. And so we would make this structure here, with two amino groups having been protonated and now shown as NH3+. An exam question could ask you to draw the structure of a particular amino acid, for example phenylalanine, at a high pH value. And they could phrase it by asking specifically about pH 14 or simply by describing it as being in an alkaline solution. When we have a high pH value, there is an excess of hydroxide ions, and these ions will react with acidic groups in the amino acid. And so this will include any group that can donate hydrogen ions. And so in phenylalanine, the proton donor is the carboxylic acid group. And so in high pH, the carboxylic acid group will be deprotonated, and this will be its structure. Amino acids can have acidic or basic R groups. Aspartic acid, for instance, has a carboxylic acid side chain, and so in a high pH solution, both of these carboxylic acid groups will be deprotonated, and so this will be the structure here with two negative groups that have lost their protons. As well as reacting together to make proteins, amino acids can react in the same ways as carboxylic acids or amines, which makes sense since they contain both of those functional groups. And so you need to be flexible when answering exam questions about amino acids. For instance, the carboxylic acid group could undergo any reaction of a carboxylic acid. And the formation of esters is the preferred reaction where they do this in an exam situation. And so in this case, the amino acid would be reacting with an alcohol. And they won't usually tell you that esterification is happening. They will say something like, draw the product formed when a named amino acid reacts with methanol and concentrated sulfuric acid. It's also worth noting that serine has both an alcohol group and a carboxylic acid group, and so technically serine could even form an ester with itself. Amino acids can react in the same way as amines too. For instance, they undergo nucleophilic substitution reactions. And a particularly common question type is a reaction of an amino acid with a halogenoalkane. And this could lead to multiple substitution, remember, if the halogenoalkane is in excess. And in this case, we would produce a quaternary ammonium salt. Both of the functional groups could also undergo nucleophilic addition-elimination reactions, either as the nucleophile in the case of the amine, or as the group being attacked in the case of the carboxylic acid. You also need to be mindful of the reactivity of the R group, this side chain. I've already mentioned serine's alcohol group side chain, but another example is phenylalanine, which has a benzene ring. And so this means that phenylalanine will react in a similar way to benzene. And so the benzene ring can undergo electrophilic substitution reactions. Proteins form when amino acids join together in a sequence to form peptide links. These reactions are called condensation reactions, since water is a product as well as the protein. During this reaction, a new bond forms between the carbon atom of the carboxylic acid group of one of the amino acids and the nitrogen atom of an adjacent amino acid. This forms what is known as a peptide link, and these have a carbonyl group adjacent to an NH group, and they can be shown in either order like I'm showing here, and the way that you would choose to show it in an exam is likely to be dictated by which way around the amino acids were given to you that you had to work out how they bonded together to form this peptide link. When a peptide link forms, the OH group from the carboxylic acid of one amino acid is removed along with one of the hydrogen atoms from the amino group in the other amino acid. And this is where the water comes from that gives this condensation reaction its name. Peptides are far smaller than proteins. They are short strings of amino acids, typically between 2 and 50 amino acids in length. Peptides have a simple primary structure based on the specific order of amino acids that they contain. You need to be able to draw the structure of a peptide formed from combining up to 3 amino acids. For instance, combining alanine with cysteine. And both of these amino acids are shown here. When this happens, a new bond forms between the carbon atom from the acid group on the alanine and the nitrogen atom on the amino group in cysteine. And water is eliminated through the loss of these 3 atoms here. And this is the dipeptide that is produced. And it's called a dipeptide because it's made from the combination of 2 amino acids. And this group of atoms here is the amide link, or it can be called a peptide link. And of course, we also produce a water molecule shown here, and this is why this type of reaction is called a condensation reaction. Alternatively, these same 2 amino acids can join in a different order to produce a different dipeptide. This time it's the acid group in the cysteine that reacts with the amino group from the alanine. And this bond forms between these atoms here, and so we produce this different dipeptide with this peptide link that I'm showing here. And also we produce water again. If you combine 3 amino acids together, you will produce a tripeptide and 2 molecules of water. For instance, if we combined alanine with cysteine and also with aspartic acid in this order, we would get a new bond forming between this carbon atom and this nitrogen, and these 3 atoms would be eliminated as a water molecule, and then we'd get a new bond forming between this carbon atom and this nitrogen, and we would eliminate another molecule of water from this collection of atoms here. And the tripeptide that we produce would look like this, and because we've combined 3 amino acids together, we will have 2 peptide links inside this peptide, and for each peptide link that we form, we will produce 1 molecule of water. And so that's why in the formation of a tripeptide, we produce 2 molecules of water. When amino acids join together to form a peptide, the amine group of one reacts with the carboxylic acid group of another, forming a peptide bond and releasing water. Even though those groups are now joined, the resulting peptide can still form a Zwitterion. Within the chain, some amino groups and some carboxylic acid groups will remain. Certainly there will be one of each at the ends of the peptide chain, but there might also be some on the side chains depending on the amino acids. These can still gain or lose protons depending on the pH of the solution. In acidic conditions, extra hydrogen ions are picked up, giving the molecule an overall positive charge. In alkaline conditions, protons are lost, giving a negative charge. At a certain pH, the peptide exists mainly as a Zwitterion, with both a positively charged NH3 plus group and a negatively charged COO minus group, but no overall charge. This balance of charge helps stabilize the molecule and affects how peptides and proteins behave in solution, including how they fold, interact, and respond to an electric field. Proteins can be referred to as polypeptides, since they can be made by joining sequences of peptides together, as well as by combining amino acids. The human body contains more than 2 million different proteins, built from only 20 different amino acids. This huge variety of proteins arises from such a small number of building blocks because of the different types of structure proteins have. And these are referred to as the primary structure of a protein, which is the sequence of amino acids it contains, the secondary structure, and this comes from localized regions of folding within the protein, and the tertiary structure, which is the overall three-dimensional shape. In an exam situation, you need to be able to recognize and identify the primary, secondary, and tertiary structures of proteins that you've been given. The primary structure of proteins is the sequence or order of amino acids present in the protein chain. The longest known protein contains more than 30,000 amino acids, so it can be a lengthy job to write the amino acid sequence for a protein, especially given that amino acids can have long, complicated names themselves. To address this issue, each amino acid is given a 3-letter abbreviation for when we write the amino acid sequence of the protein. And so, if we had part of a protein with lysine connected to phenylalanine, then connected to serine, we would write each name one after the other using a 3-letter code for each amino acid. And so this sequence of amino acids would be written like this, with lys, the code for lysine, phe replaces phenylalanine, and ser replaces serine. The links that we show between the codes indicate the peptide link between each pair of amino acids. And then on the outside are trailing bonds, which indicate that the protein chain continues in both directions after the section shown. All of this means that a long list of many amino acids looks much less complicated. Unlike peptides, which usually contain between 2 and 50 amino acids connected together, proteins can contain more than 30,000 amino acids, which is why exam questions typically ask you to draw dipeptides, tripeptides, or small sections of a protein. For example, you might be given a section of a protein, which I'm showing here, and you could be asked to draw a structure for the cis-asp section of the protein. To do this, you should start by retrieving the amino acid structures, either from the datasheet or given to you in the question. And then you should remove water from this middle here between the two amino acids, and you should form the peptide link between the carbon and the nitrogen. And now we're almost done here, but you can see this diagram is a dipeptide that I'm showing and not a part of a protein. So I need to remove water from the ends of the chain as well. This means removing one hydrogen atom from the amino group on this end and the OH part of the acid group from the other end. Last of all, you should add the trailing bonds to show that the protein is extending on in both directions. Proteins are very long three-dimensional molecules, and as a result, the amino acids in different polypeptides and in different parts of the same chain can interact, which heavily influences protein structure. And the most common type of interactions are hydrogen bonding. For hydrogen bonding to occur, there first needs to be a highly electronegative atom. All proteins contain nitrogen and oxygen, which are both very electronegative. And this makes the carbon to oxygen double bond and the NH group polar. Hydrogen bonds can form between different parts of the peptide link. Firstly, a lone pair from the oxygen atom in the carbonyl group is strongly attracted to an electron-deficient hydrogen atom from the amino group, and this attraction is the hydrogen bond. Hydrogen bonding can exist between peptide links, and this is what I'm showing in the first diagram here. We have got a polypeptide on the left-hand side, and you can see the trailing bonds on the top and on the bottom, showing that the long chain continues. And on the right-hand side, we have a second polypeptide, and on this occasion, I'm showing two hydrogen bonds between groups on opposite chains. They both start from the lone pair on the oxygen, and they both go to the electron-deficient hydrogen atom. There can also be hydrogen bonding between the R groups of two different amino acids as well. For instance, in this second diagram, I'm showing aspartic acid, which has got a carboxylic acid side chain, and I'm showing serine, which has got an alcohol as part of its side chain. And so hydrogen bonds can form like this from the lone pair on this oxygen here to the electron-deficient hydrogen atom of the alcohol. And in fact, it could also form between the oxygen of the alcohol and the electron-deficient hydrogen atom of the carboxylic acid. In exam questions, they do tend to ask more about hydrogen bonding between peptide links rather than between side chains. Another type of interaction within a protein occurs when two atoms of sulfur bond together. The amino acid cysteine contains an atom of sulfur as part of its R group, as you can see from the diagram here. Two cysteine groups from different parts of a protein can form what is known as a disulfide bridge. And you can see what happens when I draw these two cysteine groups from different parts of the same protein with their sulfur atoms facing towards each other. The hydrogen atoms are lost and a covalent bond forms between the two sulfur atoms instead, connecting them together. It's this covalent bond that I'm showing in red that is the disulfide bridge. And it's called a disulfide bridge because it uses two sulfur atoms to connect together different parts of a protein that would otherwise be separate. Since the disulfide bridge is a covalent bond, this means that disulfide bridges are stronger than hydrogen bonds, since covalent bonds are much stronger than hydrogen bonding. Ionic bonds, sometimes called salt bridges, form between charged R groups on amino acids. A good example is between aspartic acid and lysine. Aspartic acid has a carboxyl group in its side chain that can lose a proton, forming a negatively charged COO- group. Lysine, on the other hand, has an amine group in its side chain, and that can gain a proton, forming a positively charged NH3+ group. When these two side chains are close together in the folded protein, the opposite charges attract, forming an ionic bond. In an exam, you need to be able to explain how hydrogen bonding, disulfide bridges, and ionic bondings are of vital importance to maintaining the structure of proteins. And this is because these interactions influence the secondary structure of proteins as well as their tertiary structure. The secondary structure of a protein refers to how the polypeptide chain begins to fold or coil because of hydrogen bonding between atoms in the backbone, not the side chains. One of these patterns is the alpha helix, a coiled spring-like shape where hydrogen bonds form between every fourth amino acid, holding the coil tightly together. This structure is common in fibrous proteins like keratin, which gives strength to hair and nails. The other main pattern is the beta-pleated sheet, where sections of the chain lie side by side and form hydrogen bonds between them. This gives a more rigid sheet-like structure, seen in proteins like silk. What you can see from both of these diagrams is the hydrogen bonds are forming between the lone pair on an oxygen atom as part of the C double bond O group, and the hydrogen bond is going to the electron deficient hydrogen atom of part of an NH group from a different position in the polypeptide chain. So the key takeaway is that secondary structure is entirely due to hydrogen bonding between peptide bonds, creating these regular patterns that set the foundation for the protein's overall three-dimensional shape. The tertiary structure of a protein is its overall three-dimensional shape, the way the whole polypeptide chain folds and bends to produce a unique functional structure. This folding is driven by interactions between the R groups of different amino acids along the chain. We've already seen the key types of interaction: hydrogen bonding, ionic bonding, and disulfide bridges. And there are also hydrophobic interactions where non-polar side chains cluster together away from water. These hydrophobic interactions are mainly caused by van der Waals forces between non-polar groups, which hold those parts of the chain together and help keep them tucked inside the protein. These bonds and interactions occur all over the molecule, pulling parts of the chain together or holding loops and coils in place. The specific pattern of folding gives each protein its precise shape, and that shape determines its function. For example, the shape of an enzyme's active site depends directly on its tertiary structure. If these interactions are disrupted, say by changes in temperature or pH, the tertiary structure breaks down and the protein becomes denatured, losing its function completely. So the tertiary structure is all about how these smaller patterns of helices and sheets fold up into the final functional three-dimensional protein. Enzymes are proteins that act as biological catalysts. In other words, they speed up chemical reactions without being used up themselves. Enzymes have an active site, and you can see this here in our two-dimensional enzyme shown in grey, and the active site is highlighted in red. This is the region where the substrate molecule, shown here in blue, binds to the enzyme. When the substrate binds, the enzyme helps the reaction take place. For example, it might break the substrate into smaller pieces. Now, this is where the structure becomes really important. Due to the tertiary structure of an enzyme, its active site has a particular three-dimensional shape, and so the active site is referred to as being stereospecific, which means that the atoms in the active site area of the enzyme have a particular spatial arrangement, and this means that enzymes are selective. In other words, they only work on specific substrates that matches its shape. And this is known as the lock and key hypothesis. Just like a key only fits one lock, a substrate only fits one enzyme. Notice how our blue substrate has two square sections that fit perfectly into that active site. If we take a look at this second diagram, you'll see a different substrate, this time made up of yellow circles, and it doesn't fit into the active site of our enzyme at all, and so no reaction takes place. This shows how the enzyme's stereospecific shape determines which reactions it can catalyse. Some drugs can act as enzyme inhibitors, and this means that they reduce enzyme effectiveness or even stop the enzyme working entirely. The drugs block the active site, which means that the normal substrate can't bind to it, and so the reaction is blocked. This is how certain medicines slow down or stop reactions that would otherwise be harmful. In this diagram on the left-hand side, you can see the enzyme with the normal substrate occupying the active site and the reaction occurring. Meanwhile, in the second diagram, we're showing a drug, this time in red, that inhibits the enzyme action. There are a few different types of enzyme inhibitors depending on exactly how they bind, but the key idea is the same. The drug is a molecule or an ion that binds to the active site instead of the normal substrate, and since the active site is occupied, no reaction can occur. As technology improves, we can use computers to help design these drugs that block the active site of an enzyme. And the computer modelling ensures that the drug molecules are the correct shape to bind to the active site. Many enzyme substrates and drugs exist as optical isomers, which means that one molecular formula can have two enantiomers with completely different spatial arrangements. If a molecule is optically active, this means it must contain a carbon atom with 4 different groups attached to it. And this applies to all 2 amino acids, with the exception of glycine, where the R group is a hydrogen atom. As a result of this, the stereospecific active site of an enzyme can sometimes only bond to one enantiomeric form of the drug or substrate. For example, if we consider this chemical here, we can see it has a chiral center because this specific carbon atom here has 4 different groups attached to it. This means that the molecule will exist as 2 enantiomers, which are non-superimposable mirror images of each other. And so, the active site of the enzyme that interacts with this chemical will have the complementary shape to match one of the enantiomers, but not the other. And therefore, the enzyme could only break down one of these molecules. And this shows how finely tuned biological systems are. A small change in optical activity can be the difference between a reaction working perfectly or not happening at all. It's possible to break the peptide link in a protein or peptide in a reaction that's called hydrolysis. This breaks the peptide link and produces the constituent amino acids in a reaction that is the direct reverse of the condensation reactions where peptides form. In the word hydrolysis, lysis refers to breaking the peptide bond, and hydro means adding water to reform the amino acids. But you wouldn't normally do this literally by adding water. The best reagent to use is concentrated hydrochloric acid, and you would carry this reaction out by refluxing the reaction mixture at a high temperature. Or, if you prefer, you could hydrolyze using the enzyme protease and warming the reaction mixture. You need to be able to draw the structure of amino acids formed from the hydrolysis of a peptide. And whenever you do this, it will always follow the same steps, where a protein or a peptide is converted back into its constituent amino acids. For instance, if we had this tripeptide here and reacted it with concentrated hydrochloric acid under reflux, we would break these 2 peptide bonds between the carbon atom and the nitrogen atom in each case. And you can see that we now have these 3 amino acids, and the peptide bond is no longer there, and we have added OH onto the carbon atoms and another hydrogen atom onto the nitrogens. So we have effectively added water to replace each of these peptide bonds. Thin layer chromatography, often abbreviated to TLC, can be used after a protein has been hydrolyzed to separate and identify the amino acids present in the resulting solution. The most common stationary phase used in TLC is silica gel, which is highly polar. To carry out a TLC, you'd mark a pencil baseline onto the plate about 1.5 centimeters up from the bottom. Then we would use a capillary tube to add a very small drop of the solution of amino acids to the middle of the pencil line and allow the spot to dry completely. Next, add the solvent to a beaker or a developing tank, and it should be about 1 centimetre in depth, and this will ensure that the solvent is below the pencil baseline and our sample. If we're using a silica gel stationary phase, we should use a non-polar solvent, because one of the phases should be polar and the other should be non-polar. Then you would place your plate into the solvent, and we would allow the solvent to rise up the plate. It needs to reach towards the top, but doesn't have to go all of the way up. We would then remove the plate from the solvent and quickly mark on the solvent front with a pencil before the solvent can evaporate. And finally, we would allow the plate to dry. During TLC, we should use a lid or cover to prevent the solvent from evaporating, which means that the atmosphere in the beaker would be saturated with the solvent, and this will reduce evaporation of the solvent from the plate. Two important safety measures when carrying out TLC would be to wear plastic gloves in order to prevent contamination from hand to plate, and also to allow the plate to dry in a fume cupboard, since the solvents used are commonly volatile, which means they will evaporate easily, and the solvents can be toxic. Individual amino acids present in a mixture can be identified from their RF values. However, amino acids are colourless, and so they don't stand out against the white of the plate. And this means that in order to see the amino acids on the plate, we need to either spray the plate with a developing agent, such as ninhydrin, or visualise it using ultraviolet light. Having done this, before we can calculate the RF value, we must measure the distances from the pencil baseline to the spot for the sample, always measured to the middle of the spot, and also up to the solvent front line. And so it's important to mark on the solvent front in pencil quickly before the solvent evaporates from the plate. To calculate the RF value, we take the distance moved by the spot and divide it by the distance moved by the solvent. And so if our sample had moved 4 centimetres and the solvent had moved 10 centimetres, the RF value is 0.4. The RF value is a ratio of distances, and so the solvent doesn't need to reach the top of the plate. It's all about how far the spot moves relative to the solvent front, and it's expressed as a decimal that's effectively a percentage of the total distance travelled. Therefore, if the sample has an RF value of 0.4, this means it's moved 40% of the distance moved by a solvent. And this means that the RF value of 1 would mean that the sample has moved as far as the solvent has moved, and a value of 0 means that the sample is insoluble and hasn't moved at all. Amino acids have different RF values because they have different polarities, and this is because they have different R groups, and as a result they have different retention on the stationary phase, or we could say different attraction for the stationary phase, and they also have different solubility in the developing solvent, which is also known as the mobile phase. And the RF value is a ratio that reflects the balance of the solubility in the solvent and the retention on the stationary phase. Non-polar amino acids are those that have aliphatic or aromatic R groups. And in other words, these R groups will be a simple carbon chain, or they might contain a benzene ring. These non-polar amino acids will be more attracted to a non-polar mobile or stationary phase, and the opposite would be true for polar amino acids. Polar amino acids will have polar R groups, and this might be an alcohol group, or the R group might contain sulfur, or a carboxylic acid group, or an amine functional group. And these amino acids will be more attracted to a polar mobile or stationary phase. Aspartic acid and lysine in particular are both polar, and they can even carry charge under the right conditions, and so this will enhance their affinity for polar, mobile or stationary phases. Amino acids have different polarities because they contain different R groups, and so this means that they have different retention on the stationary phase and different affinity for the mobile phase. Some amino acids, however, could have the same RF value with a particular solvent. For instance, two amino acids could have the same affinity with the solvent, due to them both being equally polar, and so some amino acids might not separate or dissolve in a certain solvent, and so it may be necessary to use two different solvents, one after another. For instance, if you hydrolyzed a protein and then placed a spot of the mixture of amino acids onto a TLC plate, the solvent would rise up the plate. You would remove the plate from the solvent, let it dry, and visualize the amino acids with ultraviolet light or ninhydrin. Having done this, if these were your results, you would conclude that the sample contains 4 amino acids. However, if you rotate the plate 90 degrees and then place it into a second solvent that has a different polarity, we effectively have a new baseline here, and the new solvent would rise up the plate, carrying the amino acids with it. You'd let it dry and visualize the spots again. And this time you might see this pattern here of 7 spots. And we would now conclude that the sample in fact contains at least 7 amino acids. And the reason for the misleading results at first is that these 3 lower spots in the original plate were actually each 2 amino acids occupying the same space. But having done a TLC experiment now with 2 different solvents, we have successfully separated out these amino acids, which could now be identified by taking measurements and carrying out an RF calculation. DNA stands for deoxyribonucleic acid, and it's one of the most important molecules in all of biochemistry. A single strand of DNA is a polymer of nucleotides, repeating units that join together in a long chain. But DNA doesn't usually exist as a single strand. Instead, 2 complementary strands wind around each other to form a double helix, which is this iconic spiral shape that you can see here. Each strand carries a sequence of bases, and because the strands are complementary, one strand effectively acts as a template for the other. The bases face inwards, pairing specifically between the 2 strands, while the sugar phosphate backbones twist around the outside to give DNA its stable coiled structure. This elegant design allows DNA to store genetic information securely and replicate accurately, which is essential for life. The monomers that make up the polymer chain that is the single strand of DNA are called nucleotides. A nucleotide is made up of 3 parts: a phosphate ion, shown here, and this phosphate ion is bonded to 2-deoxyribose, which is a pentose sugar, and this means that it forms a 5-sided cyclic compound. We number the carbon atoms in the sugar, starting with this one here, being position number 1, and then we work our way clockwise until we get to here, and this is carbon number 5 as part of the branch. And then this deoxyribose sugar is in turn bonded to one of 4 bases called adenine, guanine, cytosine, or thymine. And so, since there are 4 different bases, that means that there are 4 different possible nucleotides, one with each of the 4 bases. A nucleotide is made from 3 parts: a phosphate ion, the 2-deoxyribose sugar, and one of the 4 bases. These three parts connect together in condensation reactions. In other words, they produce water when new bonds are formed. The phosphate ion connects to carbon number 5 of the 2-deoxyribose sugar, and the base connects to carbon in position number 1. And so now we have our nucleotide. If we have two of these nucleotides, they link together by forming covalent bonds between the phosphate group of one nucleotide, which is attached to position number 5, remember, and the deoxyribose of another nucleotide, and this time it bonds to carbon number 3. And this is another condensation reaction, so again we produce a water molecule. And this bonding would continue as more and more nucleotides join on until you have your complete, very long DNA molecule. There are 4 bases that could form part of a nucleotide: adenine, guanine, cytosine, and thymine. And these are often abbreviated to a simple capital letter: A, G, C, and T. When the base connects to the deoxyribose sugar, it always bonds through the NH group shown at the bottom of the structure that I'm highlighting here in red. The hydrogen atom is removed in a condensation reaction, and it's this nitrogen atom that is bonded to the sugar. The complementary strands of DNA are held together by hydrogen bonding between pairs of these bases, C always with G and A always with T. In this diagram, I'm showing the sugar-phosphate polymer chains of both strands of DNA in the helix as purple lines, and they are connected by hydrogen bonds between the bases which hold the two strands together. And I'm showing the correct number of hydrogen bonds that form between each base pair in the two complementary strands. You'll notice that cytosine and guanine always share 3 hydrogen bonds, while adenine and thymine share 2. Hydrogen bonding between base pairs is what holds complementary DNA strands together. The bases cytosine and guanine can form 3 hydrogen bonds, and adenine and thymine can form 2 hydrogen bonds. And so this is why C bonds with G rather than A or T. I'm showing our 4 bases here, and this squiggle is representing the rest of the DNA molecule. On the AQA datasheet, this is the order that the bases are given in. And so first I'm going to reorganize it slightly to put our base pairs next to each other. And then let's start by looking at A and T. I'm going to highlight the parts of each base that will form hydrogen bonds with their partner. These hydrogen bonds form in a very similar way to in protein and peptides. Hydrogen bonds form between different parts of the bases. A lone pair from oxygen or nitrogen atoms in one base is strongly attracted to an electron-deficient hydrogen atom from an NH or an NH2 group in the other base. Next, it will make it look easier if I rotate one of the bases slightly so that the atoms can have the lone pair on each base being near to the electron-deficient hydrogen atom that it's attracted to. And so you can see now that when we look at this, we have a lone pair on this nitrogen atom forming a hydrogen bond to this electron-deficient hydrogen, and then the lone pair on this oxygen forms a hydrogen bond to this electron-deficient hydrogen. And if we do the same thing with C and G, first highlighting the groups of atoms that will form the hydrogen bond, we can see that these atoms are lined up to form 3 hydrogen bonds, 2 starting from these lone pairs on atoms on cytosine, and 1 from this oxygen atom with a lone pair on the guanine. And so this is why adenine and thymine form 2 hydrogen bonds, whereas cytosine and guanine can form 3. An exam question about nucleotides might give you a partial diagram of a nucleotide and then have some slight mystery about these two regions here, which I'm labelling X and Y. And the question might simply ask, what are X and Y representing? And Y is representing the phosphate group. And this is testing your knowledge of the fact that DNA is made up of a backbone of sugar, phosphate as one polymer chain. And X is, of course, the base. And this is testing your knowledge that the bases are attached to the sugar part of the polymer chain. A second type of exam question might involve circling all or part of a nucleotide as part of a larger structure. So if they asked you to circle the phosphate, you would circle this group. The deoxyribose sugar, well, that would be this group that you would circle. If they said to circle the base, you would identify this bit here as being the base. If they said that they wanted you to circle the entire nucleotide, you would need to make sure that you circled the base, the sugar, and the phosphate. And the phosphate that you circle must be the one that is above the sugar, because that is considered to be the full nucleotide. And this is because when one nucleotide connects to another, the phosphate ion from one nucleotide connects to the sugar of the other nucleotide via carbon in position number 3. And so that is this carbon atom here. So the nucleotide that I've circled in this diagram is the true nucleotide that tells the story of how this polymer chain is being produced by connecting this phosphate ion in one nucleotide to this sugar in another. Platinum 2 complexes adopt a square planar geometry rather than tetrahedral, since this is more stable due to the electron configuration of the platinum ion. And square planar complexes can also display cis-trans isomerism. The most common example of this is Cisplatin, which is an anti-cancer drug. It has the formula PT-NH3-2Cl2. Which means that there are 2 neutral ammonia ligands and 2 negative chloride ligands. And the platinum ion has a +2 charge, and so therefore overall this complex is neutral. In cisplatin, the 2 chloride ligands are together next to each other. And in the stereoisomer transplatin, the 2 chloride ligands are opposite each other. And so both of these stereoisomers have the same formula, but they have a different spatial arrangement. And it is only cisplatin that is able to act as the anti-cancer drug. The transplatin has the wrong arrangement of its ligands. Cisplatin is an anti-cancer drug, and it works by preventing DNA replication. This is a molecule of cisplatin here, and you can tell it's the cis stereoisomer because the 2 chloride ligands are next to each other. After cisplatin enters a cell, a chloride ligand is replaced by a water ligand, and this is an equation for it here. You can see that there are 2 chloride ligands in the first complex, and afterwards one of them gets substituted out and replaced with water. And then the water molecule is replaced by a new coordinate bond, forming between platinum and a nitrogen atom in a guanine nucleotide in DNA. And then the remaining chloride ligand is also replaced as another coordinate bond forms between platinum and a nitrogen atom in a second guanine nucleotide adjacent to the first. You can see here the platinum still coordinately bonded to those 2 original ammonia molecules, now bonded to the 2 guanine nucleotides. Here's the guanine base and the deoxyribose sugar and the phosphate group in one strand of DNA, and then in another strand of DNA we have a new guanine nucleotide with a further coordinate bond forming between a nitrogen atom and the platinum. It's important to note which nitrogen atom from the base forms the coordinate bond to the platinum. It couldn't be this one because this nitrogen atom is bonded to the deoxyribose sugar. It can't be any of these three atoms because they form the hydrogen bonds to the cytosine base in the other strand of DNA. So it ought to be this nitrogen atom here, and that's the one that it is most commonly, but it could potentially be this one as well. These coordinate bonds stop the DNA strands from separating and replicating. You need to be aware that cisplatin is toxic, and it can cause side effects because it enters both healthy and cancerous cells. However, cancer cells absorb more cisplatin due to their higher blood supply, making them more affected. Additionally, we only use cisplatin in very small amounts to try to limit its negative effects. And so despite the risks, the benefits of treatment far outweigh them.